Might make a start if that's okay. So quite pleased to see that the next three speakers are somewhat below the median age. It doesn't seem that long ago to me that I was the young person at meetings like this, but that's not true anymore. So it gives me great pleasure to introduce Zoete, who is soon to be doctor if he behaves himself, and he's going to talk to us about cell type determination, which is one of the sort of exciting new fields in forensic DNA.
Great. Thank you for the introduction. This is joint work with a student that I supervised, Wessel Oosterman, and Kokshoorn from the NFI, and Marjan Sjerps, who was my supervisor during my PhD, which I will defend in 2 weeks from now. And today I would like to talk about the cell type determination association with the DNA donor based on a statistical model. Well, we've all seen today that statistical models can give you a false sense of security, but hopefully I can sort of re-establish faith in statistical models here. So often DNA evidence only tells you something about the relations to the source, so who donated this material, but in many cases there is a more interesting question, namely if there is a mixture profile and there is evidence that different cell types are contained in the stain, then you're interested in who donated what, because in some cases the fact that there's a match with a suspect doesn't mean as much when there are blood cells than when there are semen cells, for example. So let's start with some sort of a mock case example that I worked through. So a woman was orally sexually assaulted in an alley, and based on a witness statement, a suspect is apprehended. And the victim testifies that after the offender had ejaculated, she spat out the semen, and the suspect refuses to give a statement. So the victim points out a spot, and a sample is taken, and the results of the forensic investigation are a partial mixed DNA profile, a hemostasis test for blood that comes out negative, and 3 positive other cell type tests, RHD saliva, semen, and another test for seminal fluid. Now, in a way, this all sort of matches with the story that the victim tells, but there are still some questions here, like does the suspect match with the profile? What are other reasons to observe positive cell type tests? And we will look at this here. So what is sort of common is that these results are presented separately to the court. So they give some likelihood ratio or a probability measure that the suspect is part of the mixture profile. And then there is, they basically stating, well, the hemostasis test is for blood and it came out negative. And there are some other tests that come out positive. And these tests are associated with certain cell types like saliva or semen. And they add sort of a list of known false positives. So they might say, although this is a saliva test, we know that occasionally it comes out positive for urine, for example. And then it's up to the judge to combine this evidence. And from this report, it can be quite unclear how specific these tests are. Like if you just say, well, there were positive results for, say, urine, it's not clear whether that's common or not, and whether there are certain things that can make you believe that it is more common to see it, like the test was taken 2 weeks after the sample was taken. So I believe that there is some work that can assist the evaluation of the evidence here, and to work towards a combined report where you might say some sort of a probability that the suspect contributed semen, or that the suspect contributed another cell type material, or that the probability that the suspect did not contribute to the trace at all. So to quantify the value of such a positive cell type test, we need to know how common it is that the test comes out positive and semen is present, and also how common it is that the test comes out positive given another cell type. So many people in the forensic practitioners are quite aware that although a test might come out positive, it doesn't mean that there must be semen cells present in the crime stain, but they do believe that there's some sort of an increased belief, and you can quantify by having some sort of a database or testing your procedure to what extent it should increase your belief. So for example, part of a paper that is on which this presentation is based, we have a literature overview of many different cell type tests. So for example, for the PSA test, there is a study from 2004 that showed that almost all out of 176 samples that contained semen came out positive. But in the same study, they also test a slightly different procedure, and then it only comes out 169 out of 176. So what's the idea here? Well, if you look at the original paper, it says that the difference between these two tests was that the second set of samples was exposed to some high temperature for 2 days, which tells you a bit that if you expose a sample to high temperature for a long time, and it's more likely that it comes out negative afterwards. And then if you want to make some sort of a probability estimation to get a positive test, it might say something like this. So to get a positive test, according to these standards, it's approximately 175 out of 176, and it's slightly less for a different procedure. But there are many studies, and they all test slightly different under slightly different circumstances and in different labs. So although you can have all these studies, there's still the question how to incorporate these numbers in your own lab, and occasionally you might not even know where it has been exposed to high temperatures or something like that. So we'll come to that later, but of course you also need to know, apart from the intended cell type, which is already a bit awkward, I think, that a test is named after a certain cell type and then it comes out positive, that really suggests that the cell type that it was named after is present. But it's also known that it occasionally comes out positive for other cell types. So part of the same literature overview, for example, we looked at papers that tested this test on blood, which often came out negative, although the number of samples of which you tested is lower, which gives you, which makes you believe that this probability, well, is probably something like 0. But that's to use the probability, or to use a 0 probability within a probabilistic model gives you very little flexibility, because you might not have seen everything. So if you look at breast milk samples, then usually this test comes out positive, but there is a study that gives a positive result. So assuming that the test will always come out negative based on a limited sample size is well, not the best thing to do, because, for example, if the breast milk study only contained these 3 studies and this study was never performed, then you would be inclined to say 0, which leaves like no flexibility in your model, whether apparently it is possible to get a positive result. So that's essentially what I'm saying here. So to account for this, in the probabilistic model that we made, which is a Bayesian network here, you want to leave at least a small probability to get a positive cell type result, even if all the studies that we looked at came out negative. So these are the estimated conditional probabilities, which are based on a Dirichlet distribution with a uniform prior. And you see that even the studies that only showed all negatives have a very small probability, but also that when there are more samples that have been tested, you're more inclined to think that the PSA test will come out negative. So this is only a subset of the cell types that I've looked at, but I guess it's enough for the cause of this example. So as you might have seen in the previous table, for this case example, we've pooled all the data from literature, and that's a problem, as I said before, because the specifics from the sample might be different from the studies that were shown in the literature overview. Furthermore, the lab where this sample is tested in, for example, the Forensic Institute in the Netherlands, they might have a slightly different procedure. So it's not necessarily the case that this probability actually reflects the probability that you want for this specific lab or this specific sample. So we stated in the paper, and I would like to state it to anyone who's asking, that when you do such research, it's important to have some idea whether the literature overview reflects the probability from your own lab. Presumably just by testing it yourself, often according to the specifics of the case. But if this is too expensive, you can at least do a couple of them and see whether they sort of coincide with the literature overview. Furthermore, literature overview can direct you to some unlikely false positives, or give you some reason to extra value your own results because they've come close to other studies. So here I only show this test, but we've done 2 more, and another one was done in a study by Duncan Taylor, also in 2016. And combined, we can put all these probabilities in a probabilistic model. So the other evidence was the partial mixed DNA profile. For this example, we assume that it's the following profiles that come from another study, but just have something to work with. Sort of as a quick glance, you see that although the victim is, there's definitely strong evidence the victim is present, but there are some alleles missing and there's some extra alleles. So the combination of victim and suspect might be likely, but you have, would like to use some probabilistic model to come up with a probability. We use the Forensim package, but you can essentially use any package to come up with conditional probabilities to observe this evidence given different sets of donors. And you might get some sort of a likelihood ratio of 100 million times of that you obtain this DNA profile given that it was left by the suspect and the victim compared to the victim and an unknown person. And then if you put all this evidence together, you might get some sort of a Bayes net like this. So you're interested in who donated what, and that is breaking down into 2 questions: who donated stuff and what was donated. And here's the DNA results, and here are the cell type test. And here I've only included 2, but you can also include more, or even other types of evidence like RNA profiles. The benefit of putting it all in a probabilistic model is that you can look at way more alternatives than the one where you would look at when looking at it individually. So the likelihood ratio that I presented was based on the hypothesis suspect and victim versus victim and unknown person. But there can be many different combinations. You can think of the victim and the suspect, or victim and unknown. You can even think of only one person donating to the trace and having lots of drop-ins, which is unlikely, but you can at least consider it in the model. Similarly, for what we do not only need to consider the proposition of the victim, namely that's probably semen and saliva, but you can think of any cell type of which you have evidence how it reacts to the cell type test. And then combined, you have the whole set of all these things. And that's impossible, at least it's really hard to do this all without a probabilistic model, but this just propagates all the evidence through, and you get a result over all the evidence for all the different statements that you might encounter. And here, I've limited to 2 donors that all only donated 1 cell type, but you can think of more donors with more cell types per donor. Okay, so then if you would put in all the evidence, you put in the DNA profile that increases your belief that the suspect and the victim are present. And you have a positive test for semen, which increases your belief that semen is present. And you have a positive saliva test that suggests that both semen and saliva are present. And then together, you feel quite strongly that the suspect donated semen and the victim donated saliva. And you might get some probabilities like this. And of course, these are based on the prior distribution that we've selected here. That sort of links to what Bill was telling yesterday, that in these type of problems there is the problem that you have a prior distribution that influences the likelihood ratio. So you can not only present the likelihood ratio, because the prior distribution tells you how likely it is in the first place that semen is present, or that blood is present, or the saliva is present. So you're not working with 2 hypotheses anymore, but 2 individual hypotheses, but hypotheses have sub-hypotheses. So this only gives you an idea of what you would be able to do with having such a combined report in terms of reporting with the model. But you can also test sort of common assumptions, or see the increased the added value of doing additional tests. So for example, we tested the common assumption in these cases that the victim is present in the DNA mixture. So the moment you just state to the model, well, the victim is one of the donors, you can test whether this posterior probability that comes out of the model is substantially different or different at all from not doing this assumption. So we did this, and especially for DNA profiles where you have not that many loci, this assumption can have unwanted consequences, because the moment you state that the victim is present, in this case the victim is a female, then if semen is present, which is supported by the positive semen test, and the only possible donor of this semen is the other person, whereas if you look at all the different options, you might have been thinking, well, anyone could have been there, and it's going to be a mixture of an unknown person and another unknown person, both male. So this was not as obvious for me when I was doing this, but when I saw this, I thought, well, it's probably something wrong with my model. But then if you go through it, you sort of understand, wait a minute, because the presence of the victim, it limits the amount of alternatives for the positive semen test. Furthermore, as I said, the model also gives you the opportunity to test the additional value of performing extra tests on certain types of cell types, or certain cell types. So here we have simulated posterior probability that the suspect and the victim are present with a combination of semen and saliva, based on the confidence intervals that you can generate for the Dirichlet distribution for the different cell types. So as we saw before, there is a certain probability that we assume that PSA test might come back positive given that blood is present, but there's some uncertainty there. And the more tests you have performed, the more certain you are that the probability that you've assigned is close to the actual probability. So without doing any extra cell types themselves, but just testing what would happen if I would do more, you can test what it might do to your posterior probability of interest, or just the range of probabilities that you think reflect the truth. And I think that's something that is often overlooked, that you can do more types of sensitivity analysis that are commonly done with Bayesian networks, where you're only inclined what happens if this shifts to that, or that if this probability is slightly higher. We've actually simulated over the different outcomes, and simulated over given certain confidence intervals around cell types or probabilities. You can see what actually happens and whether it's worth the effort to do some extra tests. I guess I have some time, because I think it's interesting to look at another case example to point out some difficulties when working with this the probabilistic model. So in the previous model, there is quite strong evidence that there is a mixture between the victim and the suspect, which is a female and a male donor. So if semen cells are present, the only person that could have donated them, given that it's the victim and the suspect, would have been the suspect. But if you have positive cell type tests for cell types that are not gender related, for example, saliva and blood, we can have another profile that suggests that a combination of a suspect and the victim is present, again male and female, or two males. And we get the positive test for both saliva and blood, which increase your belief that saliva and blood are present. And first of all, that does increase your belief that the suspect donated saliva and the victim donated blood. But similarly, it also should increase your belief that it's the other way around, that the suspect donated blood and the victim donated saliva. And you can think of case examples where in which it's quite important who donated what. You can think of a fight or something, and then the one that was struck down probably donated blood, whereas the other one might have only have some saliva on there. So in these cases, the model does not assist you in distinguishing between these two, which are highly relevant for coming to a conclusion in the case. So then you must have some other background information. In Taylor, it was suggested in Taylor's paper from 2016 to sort of combine the quantity of DNA in your mixture with the visual appearance of the stain. So you can think of if it's a very big red stain somewhere, then you're more inclined to think that the major donor of the DNA profile is the one that donated the blood, and where the minor donor donated the other cell type material. But for this problem, I guess there's more things that you should think of, and it's not as easy as just looking at the visual appearance of the stain, because some cell types have a high quantity of DNA in them and some have less DNA. So I guess this is maybe direction for future research, or at least to to be aware of the limitation of the probabilistic model. But on the other hand, the probabilistic model sort of identifies this problem instead of. It doesn't mean that doing the original way, where you only report the individual reports, doesn't make this problem go away. Okay, so then summarized, there are some sort of take-home messages, which are mostly based for forensic practitioners, I guess. So the knowledge of the specificity and sensitivity of cell type is definitely necessary, not only to evaluate the evidence in the probabilistic model, but also in general when you report this without giving a probabilistic estimate of positive cell type or not. Furthermore, you should have data representing the workflow that is associated with the workflow of the sample from the crime stain, but you can use data from literature to further scrutinize your results. A probabilistic model, this one or another one, gives you the opportunity to look at all the different or way more alternative hypotheses than just the one presented by the victim. A situation where the suspect doesn't say anything, you might be inclined to think, well, then we do the suspect present with another cell type or suspect not present at all. But looking at all of them combined gives you the opportunity to see which ones that haven't been mentioned are quite likely or not. So this is sort of easy to do when the cell types that are likely present are gender specific, or when the question involved for the court has to do with gender specific cell types. In a situation in which it is not possible, you would need some other background information to combine this evidence. And there I would like to thank all of you and be welcome to answering any questions.
So, if you have distinct cell types, it seems that you should have a good estimate of the mixture ratio, which would then be quite helpful in deconvoluting the DNA mixture.
If you have
Distinct cell types,
yes,
you should be able to get a good estimate of the mixture ratio, because that's the ratio of the different cell types. That could be very helpful.
what do you mean with the ratio of the different cell types? If you have distinguished like blood and saliva, or what do you mean?
If you assume that one donor is the blood and another donor is the saliva, then those two cell types have very distinct chemical characteristics. So a chemical test can tell you the ratio between blood cells and saliva cells.
So in these cases, you need to add extra information on that. So usually these blood tests or saliva tests just show some sort of a positive sign, and they might give you a red bar if something is supposedly present, and then you might be inclined to include like the how red it is. But you could also use something like this, I guess so.
Oh, so no one is doing this kind of chemical analysis?
Not that I'm aware of directly in terms of combining it with the DNA evidence itself.
Great idea.
I think my question was, when you actually report this in casework, are you running with a two-stage process? So you report the strength of the evidence at the subsource level, and then there is a separate calculation at the source level. Is that right?
I do not report in such a way. I'm working as a student on this.
My point being is the calculation at the subsource level integrated somehow with the calculation at the source level? Because I've seen many cases where a scientist goes to court and they've got presumptive test on blood, and they'll give evidence to that the evidence is a billion times more likely if the blood came from the suspect. So my point is that if you're uncertain that it is blood, then clearly you cannot just apply the statistic at the subsource level to the source level. So really my question is, if you divide the calculation into two parts, then a court has got to make a decision on whether or not the DNA has come from the suspect before you can do the second part of the calculation. And that seems inherently unsatisfactory to me, because really the calculation should be rolled up into one.
I guess you make a valid argument to presenting them separately, to have the separate reports as in positive this, likelihood ratio this. But then if the court wants to accept that probably the suspect is present, then I think it's fair to sort of give him some tools to evaluate how likely is that he donated his blood or donated whatever else.
Yeah, I'm not disputing that. And I think this is, I think it's not, I don't find it a very satisfactory practice that we should be doing that, because you're asking the court to make a two-stage decision. And I think it'd be much better if it was just the one likelihood ratio that combined the probability of the evidence at the source level, including the DNA profile.
Okay, thank you.
So there are some newer genomics technologies for determining cell type, methylation and RNA. I've now been hearing talks about RNA to distinguish cell type for years now. Is that...
That does have some benefits, because if you test for RNA profiles, you actively test the same sample. So you test the RNA and DNA from the same sample. So you might be inclined to say there's some uncertainty here whether the sample that the cell type test will run on is actually the same sample as the DNA test. So you would definitely be possible to combine this with RNA profiles, but
These are not being used at the moment, or
RNA profiles are used, at least at the NFI, to evaluate cell type evidence, but it's not that common as these traditional cell types. Like they often, if they have enough material, they would definitely use the traditional cell type tests.
So is RNA not as good, or is it more expensive? Or?
I think it's better RNA because you can test it in one go and you do not lose some sample. But the interpretation of RNA is also still sort of unclear in many ways. So there have been some suggestions of how to evaluate the evidence of an RNA profile. They have markers that are associated with blood or with semen. But I guess most forensic practitioners are more used to using the traditional cell type tests. But there's no reason to just include RNA profiles as evidence node and use that instead of traditional cell type tests. Yeah.
Can I just comment on this? So I went to the Chartered Society of Forensic Sciences conference last week. Peter was there as well, so I don't know how much he saw. There were an awful lot of talks on the use of RNA on body fluid attribution. And there's a lot of issues over interpretation and cross-reactivity between different markers with different fluids. And also that actually you can't attribute the RNA to the DNA, you can't combine the two, because although you run them from the same sample, if you've got a DNA mixture and then you've got an RNA mixture of different body fluids, you're still in the same situation of, unless you've got the gender of different and you're looking at semen, you still can't attribute the DNA to that particular body fluid that the RNA is showing.
Yeah, I guess I was misunderstood then. So it's not that with RNA profiles you can directly associate the cell type with the donor, but you can at least be sure that you run it on the same sample, whereas with the separate ways, you do the cell type test on one part of the sample and you use some other part of the sample, and then you might be thinking, well, this is not necessarily the same sample. I've swapped the right and then on the left something like
Okay,
that. Yeah. Okay, thank you.
Lawyers make great stock of the use of RNA tests in the David Bain trial about whether they were
Can't hear.
acceptable and reliable. And
Can you speak up? We can't hear.
So the very high-profile retrial in New Zealand, David Bain, and there was a huge amount of discussion about RNA markers for cell type determination. So just from the point of view as whether it was an accepted technology or not, so that still has to come.
How did it?
I think it was accepted in the end. They decided it was better than the pathologist saying this is brain tissue. I think we need to move this along. Sorry, I know there are some other people who want to ask questions, but maybe we can discuss that. So thank you, Jacob.